Hello, everyone. My name is Samuel Alvarado. I am a biomedical sciences graduate student at OSU-CHS, and I'm going to be presenting my abstract, which is titled BDNF and heparin plasma blood samples using a commercial ELISA, measuring and standardization. So my presentation focuses on the methods of measuring BDNF using ELISA kits. So what is BDNF? BDNF stands for brain-derived neurotrophic factor. BDNF is a neurotrophin that plays an essential role in neuronal development and synaptic plasticity. It's also a nerve growth factor that is essential for cognitive function. But also increasing BDNF promotes neurogenesis, survival of neurons, increased excitatory transmission, and decreased inhibitory transmission in the central nervous system. So overall, BDNF is really important in promoting that neuronal growth, differentiation, and survival of those developing neurons. We also see it as a potential biomarker that can be used to look at changes that are happening in the brain. When we measure BDNF, we also have the opportunity to measure the precursor to BDNF, which is called pro-BDNF. So sometimes we refer to BDNF as mature BDNF, and we use that term interchangeably. So in this graphic right here, we see pro-BDNF and mature BDNF and the different purposes they serve. As you can tell by this graphic, BDNF is really important because it crosses the blood-brain barrier. So BDNF concentrations in the brain and blood are correlated, so it kind of gives us the opportunity to use peripheral BDNF found in the blood as a biological indicator of changes happening in the brain. So how do we measure BDNF? We use an enzyme-linked immunoabsorbent assay, or ELISA kit for short. It can be used to measure BDNF in the blood serum. However, information about sample processing and standards when clotting factors are present in the blood serum, those standards remain kind of scattered. So we have BDNF, it's a biological indicator of neural changes, and we need reliable ELISA measurements in heparin plasma blood samples, heparin being a blood clotting factor. So our aim is to validate and optimize the ELISA kit with respect to measurements in heparin in plasma blood samples. So what did we do? We took blood heparin treated plasma from 28 adults and we processed them using the ELISA kit for measurements of pro and mature BDNF. We used samples from substance abuse recovery patients and patients who are drug naive. So some of the problems we ran into and some of the solutions we found. So the kit was not validated for heparin treated plasma samples, so we ran a preliminary ELISA to determine the correct dilutions. There was poor standards for BDNF, so we ran another ELISA and vortex standards in serial dilution, and we found that it produced a highly viscous plasma sample, so we used extra caution when pipetting. So what did we find? We found that pre-analytical conditions were critical for serum samples. Determination of the intra-assay variation and the accuracy and yield at the BDNF ELISA kit heparin plasma samples were conducted with optimal dilutions. And we found that the linear dilution curve determined the optimal dilutions of plasma extracts, which we found was about 1 to 2 dilutions that optimized the detection level for pro-BDNF and 1 to 50 for dilution of mature BDNF. The range of sensitivity for BDNF isoforms in heparin plasma samples was accurate to the manufacturer's description, and we established the coefficient of variation between pro and mature BDNF was 5% for mature and 25% for pro. So we conclude that the BDNF ELISA kit determines heparin plasma BDNF accurately and with high reproducibility, which is really important if we want to use BDNF as a biomarker and we are able to measure it in blood serum accurately and with high reproducibility if the blood serum has heparin. So thank you so much. That was a summary of what we looked at and the data we collected. I want to acknowledge the OSU-CHS OBPR seats, Dr. Vasquez San Roman, Dr. Herath Gunasena, and the OSU-CHS Intramural Research Grant. Thank you so much. Have a good